I'd like to thank everybody for coming to listen today. I'm sorry for bringing the rain with me. And the title of my topic is Backwards Thinking. And it isn't really going back in time. It's thinking about things from a different angle. And hopefully by the end of this talk, you'll sort of understand why. Basically, this encompasses all the research that I've done over the last 2 years as a fellow. And it's using two cell-based assays in a functional method in a high throughput manner. And so I'll go ahead and get started. As we all know, neuroblastoma is the most common extracranial solid tumor. It accounts for 8% of pediatric cancers, but unfortunately, it accounts for 15% of pediatric cancer deaths. So we all know this is something that we need to improve upon. It's the most common malignancy of infancy, twice as common as leukemia, and 50% of cases will present with metastatic disease. Unfortunately, patients with high-risk disease have a high burden of therapy and continue to have an unacceptable mortality rate. There is a movement in neuroblastoma, in standard neuroblastoma treatment, towards utilizing targeted therapy. However, the use of small molecule inhibitors has been limited to primarily phase 1 and single-agent trials. In order to be more successful, more druggable targets must be found so that more therapeutic drugs may be developed against those genetic lesions. So why explore the tyrosine kinome? There's 91 tyrosine kinome genes. Part of it is just the fact that I work in the Drucker lab, and that's where it's gotten its claim to fame. But really, tyrosine kinases are enzymes that regulate growth, differentiation, proliferation, survival, and apoptosis. All of these processes can be deregulated in cancer. Also, tyrosine kinases such as ALK, the NTRK family, RET, and more recently ROR1 have all been implicated to have biologic roles in neuroblastoma. So the first of our assays, and sort of the one that I'll talk about the least in today's talk, is actually the validator. So I'll talk about the validator first. And it's the rapid assay, otherwise known as the RNAi-assisted protein target identification screen. And the way that it works is we take cells from either, in the case of the research that I've been doing, 10 neuroblastoma cell lines, or potentially low passage cell lines from patient samples. And we've also been using cells from patients with leukemia for a long time in our lab. We plate these cells on a 96-well plate with all of the known 91 tyrosine kinome genes. Each has a siRNA, or small inhibitory RNA, against that specific gene. Using a ligofectamine, the siRNA is driven into the cells, and at the end of 4 days of culture, they're plated on a colorimetric viability assay, which allows us to figure out what genes are critical for the quantification of cell viability. One of the things that's very nice about this assay is it's able to be completed in 4 days once the sample is cultured up and plated. So a couple of quick statistical definitions. The mean viability is the average of all the wells containing cells rather than the specific scrambled siRNA. That is included in the plate, and they're generally fairly consistent. But we feel like the mean viability actually gives us a better and more consistent value for comparison. Significant targets are defined as a decrease in the percent viability and its confidence interval more than 2 standard deviations below the mean viability. Subtle targets are defined as decreased percent viability more than 2 standard deviations. However, the confidence interval does cross the 2 standard deviation mark. And we've kept these targets because some of them are described in literature, or we've seen them be significant targets in other cell lines. So this is the example that we get for a single cell line. And you can see that this cell line, SKNAS, contains 2 significant targets, 2 subtle targets. And then in green, PLK is one that we use as the positive control because it works in almost every cell line to decrease the viability. One of the nice proofs of principle was the NRAS mutation that we saw in the cell line. And without our knowledge ahead of time, we went ahead and sequenced the cell line for NRAS mutations. We found a Q61K mutation, and then going back to the COSMIC database, found that the cell line also contains or is known to contain a Q61K mutation. And then this is the cell line that I want you to pay a little more attention to, because this is the one that I'm going to focus on more through the rest of the talk. The most significant target we saw in this was CSF1R, but we also saw important decrease in viability in the ephrin family as well. So here are the results of all 10 cell lines compiled. And all of the novel targets that really have never been described in neuroblastoma are highlighted in red. IGF-1R is known to play an important role in neuroblastoma. So CSF1R, the ephrin family, TXK, ROR1 are all novel targets within neuroblastoma. Then using the second assay, and this is the one that I intend to focus on a lot more today, is using a kinase inhibitor screen. And so the way that this works is very similar to the first. We take our cell lines or cells in culture, plate them out on a 96-well plate. Each plate in total, there are 66 different kinase inhibitors in 4 different drug concentrations. This allows us to calculate a dosing curve. At the end of 3 days of culture with each cell line, with each drug, a cell viability assay is performed. And this allows us to calculate the IC50 for that cell line. On this panel, there are 65 small molecule inhibitors. 10 are FDA approved, 35 are currently in FDA or in clinical trials, and about 20 or so are never, or at this point, are not going to be developed for further therapeutic use, but they're very helpful in figuring out targets and are considered tool compounds. An IC50 is calculated for each drug for each cell line and compared to a median IC50 that's known for each drug. This median IC50 has been derived from over 100 different cancer cell lines, including leukemia, colon, breast cancers, rhabdomyosarcoma, so not just adult cancers, a number of pediatric cancer cell lines as well. And we feel that this gives us a better comparison than trying to derive an artificial normal control. So here's what sort of comes out as a heat map that, and I'll take a second to walk you through this. On the left is each of the 10 cell lines. On the right is whether or not they're MEK-N amplified. Across the bottom is each of the 65 different compounds. And then across the top is each of the families that these compounds generally belong to. You can see that the higher, or the darker red each square shows that the more sensitive that cell line is to each of the given individual drug. So the easiest way to sort of think about this, or the most basic, is what drug works great in all 10 cell lines, or works pretty well? And the answer there is sorafenib. It works pretty well in all 10 cell lines, and maybe it's an ideal therapeutic. But if we think that way, then we started thinking of this as typical standard chemotherapy. What one drug works best for one patient. And if we consider these cell lines like patients, we can see that maybe a combination of drugs like dasatinib, vendatinib, and tizastrib might work better in one patient, highlighted here in yellow. But in another patient or another cell line, a combination of tyrosine kinase inhibitors might work best, including sorafenib, CAR-265, and CAR-258. Here's where the backwards thinking comes in. Or can we actually, instead of just figuring out what drug is best for a patient, can we actually use the drugs to think about what targets actually actively involved? One of the things to remember about these drugs is targeted therapy is still relative, even in these drugs. There are drugs like lapatinib, which has 1 or 2 targets. Sorafenib, which has a few more. Each of them have varying potency. And then there are drugs that are like storosporin, which basically is like bleach. If we take those 3 drugs that we saw in IMR 32, and we use Venn diagrams, which we all learned in kindergarten, and we use them to overlap what targets are common to all 3 drugs, we come up with 3 targets that all have something in common with all 3 drugs. That being CSF1R, KIT, and RET. And if you remember back to the first assay, we saw in this cell line target in CSF1R. And I'm going to sort of leave that one there because that's still to come out in publication. But there is correlation between both the rapid assay and the drug targeting that we're using. So remember, targeted therapy is relative. Each of these, and basically what I'm trying to get at is that it gets very complex very fast. There are 65 drugs. There are 500 kinase targets with the small molecule inhibitors. And each target has a differing potency for each drug. So the big circles represent the most potency, little circles, little potency for each target. And you need a computer to figure this all out. So we've developed a computer-based algorithm that utilizes a tier-based scoring system to predict the likeliest target for all of these greater than 500 kinase genes. If a drug should be responsive and it's not, it actually earns negative points. So it allows us to filter out all of the genes that may not be important. And so if we go back to the cell line IMR32, we are able to generate all 500 kinase genes, but throw away 200, keep 300 of them. And in the top 10% of still doing this just with the simple 3, we still end up with CSF1R, KIT, and RET. So, in conclusion, we've performed an RNAi screen of the tyrosine kinome in 10 neuroblastoma cell lines. CSF1R, TXK, ROR1, and the ephrin family all represent previously undescribed possible gene targets in neuroblastoma. There is correlation between the targets found by the siRNA screen and the small molecule inhibitor panel. And sort of where to take this in the future and how to take this from cell lines to the bedside or to the patient. Our assays are becoming potential additional diagnostic, sorry, I'm just getting over a cold. Additional diagnostics in cancer treatment. Currently at OHSU, we're about hopefully only a month away from opening a relapse and refractory AML trial that will utilize the drug screen to select a single drug or a combination of drugs that will hopefully be best in treating these patients with AML. Ideally, these two assays can be utilized in patients with solid tumors directly or generating low passage cell lines from patient samples. Eventually, the small molecule assay can be used to design combination therapy. I'd like to take this opportunity to thank my mentor, Dr. Drucker, Jeff Tyner, and Bill Chang, to thank other members of our lab, my clinical mentor, Suman Malampati, most importantly, the audience, the parents here, all the other researchers, and then lastly, I'd like to thank my funding source, which has been Emily's Power for a Cure, which is a parent-based organization.